ये है बायोमोलिक्यूल का एनसीईआरटी का सॉल्यूशन। तो इसमें कि ग्लूकोज और द सुक्रोज आर soluble in the water, but cyclohexane or the benzene, simple six-membered rings are insoluble in water. Explain. The glucose molecule contains the 5 OH group, while the sucrose molecule contains 8 OH groups. Thus, the glucose and the sucrose undergo the extensive H bonding with the water. Hence, these are soluble in the water. But cyclohexane and the benzene do not contain the OH group. Hence, they cannot undergo the H bonding with the water, and the result these are insoluble in the water. Next, what expected product of the hydrolysis of the lactose? The lactose is composed of the beta-D-galactose and the beta-D-glucose. From the hydrolysis, it gives the beta-D-galactose and the beta-D-glucose. तो ये हो गया beta-D-galactose, ये हो गया beta-D-glucose. तो लैक्टोज प्लस दे देगा डी प्लस ग्लूकोज और डी माइनस ग्लैक्टोज। फिर आता है कि हाउ डू यू एक्सप्लेन द एब्सेंस ऑफ द एल्डिहाइड ग्रुप इन द पेंटा एसिटेट ऑफ द डी ग्लूकोज? तो डी ग्लूकोज रिएक्ट विद द हाइड्रोक्सिलामाइन टू फॉर्म एन ऑक्साइड बिकॉज ऑफ द प्रेजेंस ऑफ द एल्डिहाइडिक group or the carbonyl carbon. This happens when the cyclic structure of the glucose forms an open chain structure of the aqueous medium, which then react with the NH2H to give an oxime. तो ये हो गया ग्लूकोज NH4 के साथ रहेगा तो ऑक्साइड बनाता है, बट पेंटाफिटेट ऑफ द डी ग्लूकोज डे नॉट रिएक्ट विद NH2H, दिस इज बिकॉज द पेंटाफिटेट डे नॉट फॉर्म एन ओपन चेन स्ट्रक्चर। तो ये हो गया ग्लूकोज ओपन चेन फॉर्म, इससे बनेगा अल्फा ग्लूकोज, इससे बनेगा ठीक है ग्लूकोज ऑक्साइड। ये हो गया अल्फा ग्लूकोज बीटा ग्लूकोज। इधर पे आता है कि अल्फा ग्लूकोज पेंटाफिटेट और बीटा ग्लूकोज पेंटाफिटेट, नो एग्जाम ठीक है, यहाँ कोई एग्जाम है जो नहीं बनेगा। अगला सवाल द मेल्टिंग पॉइंट फॉर द सॉल्युबिलिटी इन द बटर ऑफ द अमीनो एसिड आर जनरली हायर देन दैट ऑफ द corresponding halo acid. सही है। तो both acidic as well as the basic groups are present in the same molecule of the amino group. In the aqueous solution, the carboxyl group can lose a proton and the amino group can accept a proton, thus giving rise to a dipolar ion known as the deuter ion. So due to the dipolar behavior, they have the strong electrostatic interaction between them and with water, but halo acid do not exhibit such dipolar behavior. For this reason, the melting point and the solubility of the amino acid in the water is higher than those of the corresponding halo acid. Where does the water present in the egg go after boiling the egg? So when an egg is boiled, the proteins present inside the egg get denatured and coagulate. After boiling the egg, the water present in it absorbed by the coagulated protein through the H bonding. Next, why cannot vitamin C be stored in our body? So vitamin C cannot be stored in our body because it is water soluble. As a result, it is readily excreted in the urine. Next, what products would be formed when a nucleotide from the DNA containing the thymine is hydrolyzed? So when a nucleotide from the DNA containing thymine is hydrolyzed, thymine, beta D2 deoxyribose and the phosphoric acid are obtained as the product. Next, when the RNA is hydrolyzed, there is no relationship among the quantity of the different bases obtained. So what is the risk factor suggest about the structure of RNA? So a DNA molecule is double stranded in which the pairing of the base occur. Adenine always pair with the thymine, while the cytosine always pair with the guanine. Therefore, on the hydrolysis of the DNA, the quantity of the adenine produced is equal to that of the thymine, and similarly, the quantity of the cytosine is equal to that of the guanine. But when the RNA is hydrolyzed, there is no relationship among the quantity of the different bases obtained, hence RNA is single-stranded. Next, what are the monosaccharides? So monosaccharides are the carbohydrates that cannot be hydrolyzed further to give the simpler units of the polyhydroxyaldehyde or the ketone. Monosaccharides are classified on the basis of the number of the carbon atoms and the functional group present in them. Monosaccharides containing an aldehyde group are known as the aldolases, and those containing a keto group are known as the ketoses. Monosaccharides are further classified as the trioses, tetroses, pentoses, hexoses, and the heptoses according to the number of the carbon atoms they contain. For example, a ketose containing 3 carbon atoms is called the ketotriose, and aldose containing 3 carbon atoms is known as the aldotriose. फिर आता है भाटे reducing sugar. तो reducing sugar for carbohydrate that reduce the failing function and tolerance agent. All monosaccharides and disaccharides excluding the sucrose are the reducing sugars. Next, write two main functions of the carbohydrate in the plant. The two main functions of the carbohydrate in the plant are polysaccharides such as the starch formed by the storage molecule, or cellulose. A polysaccharide is used to build the cell wall. फिर आता है classify the following into the monosaccharides and the disaccharides. तो राइबोज, 2-deoxyribose और लैक्टोज या फ्रुक्टोज ये मोनोसैकेराइड है, माल्टोज या लैक्टोज जो है डाइसैकेराइड है। नेक्स्ट है व्हाट डू यू अंडरस्टैंड बाय द टर्म ग्लाइकोसिलिक लिंकेज? तो ग्लाइकोसिलिक लिंकेज refers to the linkage formed between two monosaccharide units through the oxygen atom by the loss of the water molecule. For example, if in a sucrose molecule, two monosaccharide units glucose and the beta fructose are joined together by the glycosidic linkage. ठीक है। नेक्स्ट what is glycogen? How it different from the starch? तो glycogen is a carbohydrate polysaccharide. In animals, carbohydrate are stored as the glycogen. Starch is a carbohydrate consisting of two components, amylose 15 to 20%, amylopectin 80 to 85%. However, glycogen consists of only one component, which structure is similar to the amylopectin. Also, glycogen is more branched than the amylopectin. Next, what are the hydrolysis products of the sucrose and the lactose? So on the hydrolysis, sucrose gives one molecule of the डी ग्लूकोज एंड 1 मोलेक्यूल ऑफ द बीटा डी फ्रुक्टोज। तो ये हाइड्रोलिसिस करेंगे तो अल्फा डी ग्लूकोज और बीटा डी फ्रुक्टोज मिलेगा। फिर द हाइड्रोलिसिस ऑफ द लैक्टोज गिव द बीटा डी लैक्टोज एंड द बीटा डी ग्लूकोज। तो बीटा डी लैक्टोज प्लस बीटा डी ग्लूकोज मिल जाएगा। नेक्स्ट है what is the basic structural difference between the starch and the fulose? The starch consists of the two components, amylose and amylopectin. Amylose is a long linear chain of the D-glucose units joined by the C1C4 glycosidic linkage. Amylopectin is a branch chain polymer of the D-glucose units in which the chain is formed by the C1C4 glycosidic linkage and the branching occurs by the C1C6 glycosidic linkage. ये हो गया amylopectin. On the other hand, cellulose is a straight chain polysaccharide of the beta-D-glucose units joined by the C1C4 glycosidic linkage. Beta links होता है. Next, okay, what happens when the D-glucose is treated with the following reagents: HI, bromine, butter, HNO3. तो when the D-glucose is treated with HI for a long time, NHN जो फॉर्म होता है, D-glucose HI हिट करेंगे तो बनेगा NHN. When D-glucose is heated with Br2, D-gluconic acid ही प्रोड्यूस होता है. तो Br2 बाटा के साथ करेंगे तो D-gluconic acid होगा. On being heated with HNO3, D-glucose gets oxidized to give the saccharic acid. तो D-glucose HNO3 के साथ बनाएगा saccharic acid. Next, enumerate the reactions of the D-glucose which cannot be explained by its open chain structure. So aldehydes give the 2,4-DNP test, skip test, and they react with the NH4 to form the hydrogen sulfide addition product. However, glucose does not undergo this reaction. The pentaphytate of the glucose does not react with the hydroxylamine. This indicates that the free switch group is absent from the glucose. Glucose is used in the two crystalline form, alpha and the beta. The alpha form melting point of nice Kelvin crystallized form a concentrated solution of the glucose at 1014 Kelvin, and the beta form crystallized from a hot and the saturated aqueous solution of the 370 Kelvin. This behavior cannot be explained by the open chain structure of the glucose. Next, what are the essential and the non-essential amino acids? Give two examples of each type. The essential amino acids are required by the human body, but they cannot be synthesized in the body. They must be taken through the food, for example, valine and leucine. Non-essential amino acids are required by the body, but they can be synthesized in the body. For example, glycine and the alanine. फिर आता है कि define the following related terms to the protein: peptide linkage, primary structure, and the denaturation. So peptide linkage. The amide formed between the feed allergy group of the one molecule of amino acid and the NH2 group of another molecule of amino acid by the elimination of the butter molecule is called the peptide linkage. तो ये हो गया. ठीक है. Valine, adenine, vala, al. Primary structure. The primary structure of a protein refers to the specific sequence in which the various amino acids are present in it. The sequence of the linkage between the amino acid in the polypeptide chain. This sequence in which amino acids are arranged different each in the each protein. A chain in the sequence creates a different protein. Denaturation. In a biological system, a protein is found to have the unique three-dimensional structure and a unique biological system. In such a situation, the protein is called the native protein. However, when the native protein is subjected to physical change such as the change in the temperature or the chemical change such as the change in the pH, its H bond are disturbed. This disturbance unfolds the globules and uncalls the helix. As a result, the protein loses the biological activity. This loss of the biological activity by the protein is called the denaturation. During the denaturation, the secondary and the tertiary structure of the protein get destroyed, but the primary structure remains unaltered. One of the examples of the denaturation of protein is the coagulation of the egg white when the egg is boiled. फिर आता है कि what are the common types of the secondary structure of the protein? So there are two common types of the secondary structure of protein: alpha helical structure, beta pleated structure, or helical structure. So in this structure, the NH group of an amino acid residue forms the H bond with the food allo group of the different atom are turned to form the right-handed screw helix. The beta pleated suite. This structure is called so because it looks like the pleated folds of the drapery. In this structure, all the peptide chains are stretched out to the nearly maximum extension and then laid side by side. These peptide chains are held together by the intermolecular hydrogen bonds. फिर आता है कि what type of the bonding helps in the stabilizing the alpha helical structure of the protein? The H bonds formed between the NH group of the each amino acid residue and the pseudo group of the adjacent terms of the helix in the stabilizing the helix. फिर आता है differentiate between the globular and the fibrous protein. तो ये हो गया fibrous protein और ये globular protein. Globular protein में होता है कि ये fiber like structure formed by the polypeptide chain. These proteins are held together by the strong hydrogen and the disulfide bonds. It is usually insoluble in the butter, or fibrous proteins are usually used for the structural purpose. For example, keratin is present in the nails and the hair, collagen in tendons, and myosin in the muscles. Globular protein में आता है कि the polypeptide chain is the protein is folded around itself, giving rise to the structural spherical structure. It is usually soluble in the butter. All enzymes are globular proteins. Some hormones such as the insulin are also globular proteins होते हैं. फिर आता है कि how do you explain the amphoteric behavior of the amino acid? In the aqueous solution, the carboxyl group of an amino acid can be lose a proton, and the amino group can accept a proton to give a dipole ion on the deuterion. Deuterion. Therefore, in the deuterion form, the amino acid can act both as the acid and the base. Thus, amino acid for the amphoteric behavior. फिर आता है what are enzymes? The enzymes are the proteins that catalyze the biological reaction. They are very specific in the nature and catalyze on a particular reaction of a particular substrate. Enzymes are usually named after the particular substrate or the class of the substrate and even sometime after the particular reaction. For example, the enzyme used to catalyze the hydrolysis of the maltose into glucose named as the maltase. तो maltase से हो गया glucose. Again, the enzyme used to catalyze the oxidation of one substrate with the simultaneous reduction of the another substrate and another oxidoreductive enzyme. The name of the enzyme ends with the H. What is the effect of the denaturation of the structure of the protein? As a result of the denaturation, globules get unfolded and helix get uncoiled. Secondary and tertiary structure of the protein are destroyed, but the primary structure remains unaltered. It can be said that during the denaturation, secondary and tertiary structure proteins get converted into the primary structure protein. Also, as the secondary and tertiary structure are destroyed, the enzyme lost its activity. How are vitamins classified? Name the vitamin responsible for the coagulation of the blood. So on the basis of the solubility in water or fat, vitamins are classified into two groups. Fat soluble vitamins. Vitamins are soluble in the fat and the oils but not in the butter. Belong to this group. For example, A, D, E, K. Butter soluble vitamins. Vitamins that are soluble in the butter belong to this group. For example, group B vitamins B1, B2, B6, B12, etc. and vitamin C. However, biotin, the vitamin is neither soluble in the butter nor in the fat. Vitamin K is responsible for the coagulation of the blood. By vitamin A and the C, vitamin C is essential to us, give their important forces. The deficiency of vitamin A leads to the geothermia, hardening of the cornea of the eye, and the night blindness. The deficiency of the vitamin C leads to the scurvy, bleeding gums. The source of the vitamin A are the fish, liver oil, carrot, butter, and the milk. The source of vitamin C are the citrus, amla. ये सब हो जाता है. What are nucleic acids? Mention that two important functions. Nucleic acids are the biomolecules found in the nuclei of all living cells and is one of the constituents of the chromosome. There are mainly two types of nucleic acid: DNA or RNA. Nucleic acids are known as the polynucleotides and they are long-chain polymers of the nucleotide. Two main functions of the nucleic acid are DNA is responsible for the transmission of the inherent characters from the one generation to the next. This process of transmission is called the heredity. Nucleic acid both are responsible for the protein synthesis in a cell, even though the proteins are actually synthesized by the very far in the molecule in the cell. The message for the synthesis of particular protein is in the DNA. What is the difference between nucleoside and the nucleotide? The nucleoside is formed by the attachment of the base of one position to the sugar. Nucleoside provides the sugar plus base. On the other hand, all the three basic components of nucleic acid, pentose, sugar, phosphoric acid, and the base are present in the nucleotide. Nucleotide is equal to the sugar plus base plus phosphoric acid. Yoga structure of a nucleotide. Next, the two strands of DNA are not identical but are complementary explained. So, in the helical structure of DNA, the two strands are held together by the hydrogen bonds between the specific pair of bases. Cytosines form the hydrogen bond with the guanine, while the hydrogen bond with the thymine. As a result, the two strands are complementary to each other. What is the important structure and the function between the DNA and RNA? So, the structural difference between DNA and RNA is to say the sugar moiety in the molecule beta D2 deoxyribose. DNA contains the thymine, it does not contain the uracil. हेलिकल स्ट्रक्चर ऑफ डीएनए डबल स्ट्रैंडेड होता है. यहाँ पर आता है सुगर मोटिन द डीएनए मॉलिक्यूल बीटा डी राइबोज. आरएनए कंटेन सुगरफिल डज नॉट कंटेन द थायमिन और द हेलिकल स्ट्रक्चर ऑफ द आरएनए सिंगल स्ट्रैंडेड होता है. द फंक्शनल डिफरेंस बिटवीन डीएनए आरएनए है डीएनए और आरएनए is the chemical basis of the heredity. DNA molecule do not synthesize the protein but transfer coded message to the synthesis of the protein in the cells. Whereas the RNA is not responsible for the heredity or proteins are synthesized in the RNA molecule in the cells. What are different types of the RNA found in a cell? तो मैसेंजर RNA, रिबोसोमल RNA और ट्रांसफर RNA, tRNA आते हैं. ठीक है, तो यहाँ पर है जो इसका सॉल्यूशंस कंप्लीट होता है.